Every year, I make resolutions. Resolution is a great word with so many different meanings. 1. A firm decision to do or not do something. Let's resolve to discuss vault structure. 2. The quality of being determined. We were resolute in our intention to determine vault structure. 3. The action of solving a problem, dispute, or contentious matter. The images gave a resolution to the problem. 4. The process of reducing something into its components. Our methods showed a high degree of detail, a high resolution. 5. The conversion of something abstract into another form. Our uncertainty gradually resolved into understanding. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist with a passion for vaults. My daughter-in-law Tara and I are making videos to explain what vaults are to non-scientists. In our last video, we showed you some low-resolution structures of the vault and how we determined that the entire outer shell of the vault was made from a bunch of copies of just one protein, the major vault protein. If you haven't seen it, I'll put a link below. Today, I want to cover 3 things. First, I want to remind you how we determine the basic structure and composition of vaults and pick up today's theme, resolution. Second, I want to introduce you to a high-resolution technique called X-ray crystallography. And third, I want to show you how we spent 3 years determining the atomic structure for a vault, and how, completely unbeknownst to us, a Japanese scientist working on vaults labored for 5 years to solve an even higher resolution structure. Do you think our model for the position of MVP in the vault shell was correct? Stick around and you'll find out. In the last 2 videos, I introduced cryo-EM reconstruction, and I showed you lots of vault reconstructions that were carried out on thousands of particle images, giving moderate resolution, but still relatively low in the neighborhood of 20 angstroms. Here's a model I made of the 20 angstrom resolution vault. Although cryo-EM was getting better, when this model was produced, we couldn't see individual MVP protein chains. In order to pick out individual amino acids, we needed to get to a resolution of 10 angstroms or less. To get to this resolution, we turned to X-ray crystallography. I'll take a minute to give you a simplified explanation of the technique. We touched on X-ray crystallography in episode 7 when I mentioned Rosalind Franklin's X-ray data that allowed Watson and Crick to propose the structure of DNA. This technique allows one to determine the atomic and molecular structure of a crystal. X-ray crystallography originated a few years after X-rays were discovered in 1895 by Wilhelm Conrad Röntgen, a German scientist. He described a form of radiation of unknown nature, hence the name X-rays. This radiation could penetrate opaque bodies. He used the radiation to take the first X-ray. It was a picture of his wife's hand. Röntgen was awarded the Nobel Prize in 1901 for his discovery. To perform X-ray crystallography, a beam of X-rays is sent through a crystal, and it diffracts into many different directions. By measuring the angles and intensity of these diffracted beams, a crystallographer can produce a 3-dimensional picture of the density of electrons within the crystal and determine the positions of the atoms in the crystal. To carry out X-ray crystallography on a protein or a particle, it requires that the object be grown as a fairly large crystal. The bigger the structure, the more difficult it is to grow large crystals of that substance. In addition, growing a crystal requires a very pure sample. Nevertheless, in 2004, we initiated a collaboration with David Eisenberg's lab at UCLA to attempt to grow a crystal of vaults and obtain a high-resolution structure. David Eisenberg is a colleague and friend who happens to be one of the most accomplished structural biologists in the world. David asked Dan Anderson, a skilled crystallographer in his lab, to work with Val on growing a crystal. And after 3 years of various attempts, Dan got a few small crystals to grow from one of the recombinant vault preparations that Val made in insect cells. These crystals were barely large enough to get some X-ray data. However, Dan packed up the crystals and drove up to the Advanced Light Source at the Lawrence Berkeley National Laboratory. The Advanced Light Source, nicknamed ALS, is a resource of the Department of Energy. ALS is a specialized particle accelerator called a synchrotron that generates bright beams of X-ray light for scientific research. At ALS, electrons are generated and accelerated in a linear accelerator. They are then bent with magnets and directed into a circle and further accelerated to 99.999% of the speed of light. They are stored in a bigger outer ring where they can be manipulated to generate all kinds of particles, including X-rays. The X-rays are directed out of the storage ring through beam lines to experimental end stations where scientists can conduct research. For example, X-ray crystallography of biological samples. ALS originally cost about $100 million to build. I think it was worth every penny. Dan was able to get enough data to generate a moderate 9 angstrom resolution structure of the vault. The structure he obtained in 2007 confirmed our model that vaults had 2 identical halves divided at the waist, and that MVP was distributed along the length of a half vault running from its end terminus facing the inside of the vault at the waist up to the cap. There was one thing in Dan's structure that we got wrong. Dan predicted 96 copies of MVP per vault. However, this number was partially the result of the 48-fold averaging that Dan imposed in order to optimize the resolution. This number, 96, fit our previous model and explained the 8-petal flowers generated by John Heuser as being composed of maybe 6 MVPs per petal, so we thought it might be correct. When Dan's structure was published, it was not featured on the journal cover. However, just this past summer, the magazine of the Royal Society of Biology, The Biologist, wrote a feature article about vaults and recognized the beauty of the particle with a 2-color version of Dan's vault. Let's do our cover magic. 2 years later, in 2009, a paper was published in the journal Science describing an even higher resolution structure of the vault by a group in Japan headed up by Hideki Tanaka. We didn't even know Professor Tanaka was working on vaults, but it turned out he spent 5 years growing a crystal of rodent vaults that he purified using our published protocol. Hideki was able to get a 3.5 angstrom structure that was very similar to Dan's structure. However, Hideki found that there were only 78 copies of MVP per vault, or 39 in each half vault. Hideki's crystal structure is shown here. All of the MVP chains are shown in what is called a ribbon form. One chain is colored red in the vault, and a copy of that MVP is shown to the left of the vault. This is the MVP chain that I used in my last video when I explained our cryo-EM studies to you. Hideki's structure and a more recent high-resolution cryo-EM reconstruction allowed me to print this vault on my 3D printer. I painted one of the vault chains black so you could see how it is positioned in the structure. So on the one hand, I felt great that our earlier cryo-EM structural studies got the orientation of MVP correct. However, I was confused with the finding that there were only 78 MVP chains per vault. Don't get me wrong, I believed the data. The problem was that at that time, we thought that vaults assembled into halves, and that the two halves came together to form the whole vault, and I just didn't understand how nature could work in multiples of 39. It took us 10 years to solve that mystery. I'm Lenny Rome, the Vault Guy. I hope you join me in my next video, where I tell you about our experiments that led to a unique solution to vault assembly. Please remember to subscribe and like, and feel free to leave a comment.